Good afternoon to all the EAPCET aspirants. So here we are with the EAPCET Previous Year Questions from the first chapter of plant physiology, that is transport in plants, after which we are going to look into mineral nutrition. So here you see, choose the correct statements, correct set of statements from transport in plants. First statement based question, look into that carefully ma. First statement, porins allow the entry of small proteins present in the membrane of mitochondria, chloroplast and bacteria. Yes, of course, this is the correct statement. Porins are what? Porins which will allow small proteins that are present in the membrane, outer membrane of mitochondria, chloroplast and bacteria. That is true. When a living cell is kept in hypertonic solution, water potential is equal to solute potential. Wrong. Okay, water potential is not equal to solute potential when a living cell is placed in hypertonic solution. What happens when a living cell is placed in a hypertonic solution? Look at here carefully. Let us suppose this is a hypertonic solution into which a cell is placed. After a certain period of time, what happens is water molecules will move away from the cell, as it is the hypertonic solution. As the cell is placed in the hypertonic solution, water molecules will exit out of the cell and the cell will shrink eventually. Okay, so here there is no equal water potential. Water potential is differing both inside the living cell and in the hypertonic solution. Fine, right? Negative water potential will be present in the hypertonic solution when compared to the living cell. That is the reason water molecules will move out of the living cell, which leads to the shrinkage of the living cell. So option B is wrong. And then if you move on to the next question, next statement, carrier proteins will allow entry of all substances across the membrane, whereas this pumps transport large molecules in passive mode. Yes, this transports large molecules in passive method of transport. It will not happen. See, carrier proteins will allow entry of all substances across the membrane, whereas pumps will transport large molecules in passive method. No, pumps will transport in active method. Porins will transport in passive method. This transports in active method. Fine, right ma. Next. Yeah. Look at this. Bulk flow occurs by diffusion. Wrong. Bulk flow doesn't occur by diffusion. Once look at the second statement ma. When a living cell is placed in hypertonic solution, water potential will be equal to solute potential. Water potentials, I felt like it is both water potential. No, water potential of both will not be equal, but water potential here, it will be equal to the solute potential which is present inside the solution. B is true only. So A and B both are correct. C and D will be incorrect. Fine. Understood everyone? Carrier proteins, they will not allow all the molecules, and pumps will transport by active transport, not by passive method. Bulk flow is not by diffusion. Clear? And then next question we will look on to, again a statement based question. Look at here to the statements carefully. First statement, concentration gradient must already be present from molecules to diffuse even if facilitated by proteins. Yes, concentration gradient, if it is present already, then only passive method of transport is possible. Next, a seed may appear dry as it has no water. Limited amount of water is present, no water is wrong. Next, the absorbance of water by corn plant is 5 liters in a day. No, it is not a 5 liters by a single plant, it will be approximately 3 to 4. Next, numerically osmotic pressure is equal to osmotic potential with change in sign. Correct? Transpiration is affected by root shoot ratio. True. So the right option here will be A, D, E, which is option B is the right answer. Clear everyone? Any doubt anywhere ma? And then look at the next question. Pickles are preserved for longer period of time by addition of salt. Which process is involved here? The process involved over here is plasmolysis. The process involved here is plasmolysis, that when they are preserved in high concentrated solutions, they will preserve for a longer time. It will not get spoiled. Fine. And then fine. Look at the next question. Next question. Yeah. Here we go, which is a match the following type of question. So all the questions till now we have seen, mostly all of them are multi-statement based questions only from this chapter. Okay, so that is how you have to exercise it. Now this question you see, which is match the following type of question. First is porins. Okay, porins. I hope the previous question is clear to everyone. Okay, here first is what ma? Porins. What does porins do? Porins, these are the proteins which form huge pores on the outer membranes of some bacteria, mitochondria and chloroplast, which is option C. Next is solute potential. Solute potential is lowering of potential due to dissolution of solute. More the solute you add, more will be the solute potential, and solute potential will always be negative. Then apoplast. Apoplast is the system of adjacent cell walls. Apoplast is the system of adjacent cell walls. The transport will be across the cells. Okay, it is in between adjacent cell walls. This is continuous throughout the plant body. Okay, which is option D. Next is scotoactive stomata. Scotoactive stomata is present in CAM plants. This is the type of stomata where stomata opens during night time and closes during daytime. Okay, scotoactive stomata, where this is usually observed in CAM plants. CAM plants where stomata opens during night and closes during daytime. Here it is option A. Here we go. First one is C, followed by B, D and A. Option C is the right answer. I hope this is clear to everyone. The question is clear to everyone, right ma? Yeah. Enough. Now we will move on to the next question, which is again a match the following type of question. Look at it carefully first. All the phenomenons are given. Imbibition. Imbibition is the diffusion of water molecules by solid. Usually we will observe the increase in size of wood during winter season, and seed germination where water is being absorbed, okay, by the dry seed for the process of seed germination. So here what is imbibition ma? Imbibition is usually a diffusion process by what? Solids. Next, guttation. Guttation will increase what? Root pressure. Next, suberin. Suberin is the depositions that you can see in the root endodermis. D is 2. Guttation is related to what? Root pressure. And then this, just a second, sorry. What is guttation? Usually in the winter mornings, if you can see, over the margins of the leaf at the lower temperatures, you can observe the water droplets that are formed. This is due to the phenomena of guttation. So where is it observed? It is observed in the shoot epidermis. Okay, this is observed in shoot epidermis. Desiccation is seen, it is because of what? Root pressure. So here guttation, it is seen in the shoot epidermis. Imbibition is observed. Imbibition is because of what? Diffusion by solids. And desiccation, root pressure, and suberin depositions are observed in the root endodermis. The option will be B. 5. Yeah. Next question. Yeah. Again a statement based question. Appeared in 2019. Few more set of questions, guys. Hold on. In pressure flow hypothesis, that is transport of sugars, these are transported from sucrose, from source to sink. The initial steps which occur are, what are the initial steps? So regarding the steps that are seen, first sucrose transport, this is the second question that we are observing, multi-statement question. This is the second question that we can see where the transport of sucrose is involved. In leaves, glucose is converted to sucrose. Yes, of course, glucose for the transport, it will be converted to sucrose. Fine, right? To be stored, glucose will be stored in the form of starch. Sucrose will move from sieve tube cells to companion cells by active transport. True? Because the concentration will be more increased in the companion cells. Fine. Loading of sucrose produces hypotonic condition in phloem. No, it will produce hypertonic condition in the phloem. Hypertonic condition. Next, water in adjacent xylem will move to phloem by osmosis. No. So here what will be the right answer? 1 and 2. Clear? Clear till here, ma? Any doubt anywhere? Yeah. Next. Again a statement based but assertion reason type of question. Okay, see, you are getting exposed to all the type of questions. Multi statement based, assertion reason. Okay, and match the following type of questions. Here we go with the assertion. Apoplastic path of water is considered to be non-living path. In apoplastic pathway, water does not move through intercellular spaces or through space between the cell wall and plasma membrane. Wrong. Apoplastic pathway and symplastic pathway, let us recall it once. If these are the adjacent cells that are being present, we know two cells are connected by what connections? Plasmodesmatal connections. And let us consider this is the cytoplasm which is inside the cell, and this is the space between the cell membrane and cell wall. Fine, right? So there are two methods of transport of water and mineral nutrients. So here if you see the transport, if it is in the space present in between cell membrane and cell wall, this transport is through the cell wall space. Okay, transport through cell wall space. Fine, right? This method of transport is the apoplastic pathway. Apoplastic pathway. And if it is through intercellular spaces, that is through cytoplasmic streaming, it is in between the intercellular spaces, that is symplastic pathway. Symplastic pathway. Fine, right? Apoplast and symplast. So assertion is true. Apoplastic path of water is considered as non-living path. True. But then in apoplastic pathway, water is moved through the intercellular spaces. Here it is saying in the reason like it does not move through intercellular spaces. So reason will be false, but assertion is true. A is true and R is false. Option C is the right answer. Next question. Assertion reason type of question again. Here, sodium chloride is dissolved in water will have low water potential than the pure water. True. Because sodium chloride, which is the solute, when it is diluted in the water, it will decrease the water potential of the pure water. Okay, in a solution, water potential will be decreased because of the dissolution of solute molecules. So, assertion is true. In reason, if you see, the solute potential is always positive. Wrong. Solute potential will always be negative because it is reducing the water potential. Solute potential is always negative in comparison with that of pure water. So, assertion is true and reason is false. A is true and R is false. Option C will be the right answer. Next question and last question of today's session. And then we will go with the chapter mineral nutrition completely. Okay, hold on students. Look at this last question which is asked in the year 2018. Which of the following requires energy? Okay, passive transport can be of three modes. One is through diffusion. Second is facilitated diffusion and third will be osmosis. All these three methods of transport, doesn't require any energy because it is across the concentration gradient. Okay, this is across the concentration gradient. This is across the concentration gradient. And next is active transport. Active transport is against the concentration gradient. So, it requires ATP energy. Fine. Clear till here, ma? So, the right answer here is active transport. Fine. Clear. And then we will move on to the chapter that is mineral nutrition. Just a second. And here we go with this chapter. Yes. From mineral nutrition, what is that we have to look into for the exam point of view? This is completely memory based chapter which will just take half an hour for you to revise. Okay. Here you have to look into what are the essential elements. What are the essential elements that are required for the plant, and what are macronutrients and what are micronutrients. Macronutrients and micronutrients, what are the deficiency symptoms that you can see in the absence of macro and micronutrients. The deficiency symptoms, macro and micronutrients, and in the beginning you have to have an idea on the hydroponics. Okay, what is hydroponics? What is the basic definition? What are we going to do with hydroponics? That is the first information that you have to know. And the second is about the essential elements, macronutrients, micronutrients, and the deficiency symptoms that are being observed in the absence of macro and micronutrients, followed by lastly nitrogen metabolism. Lastly, it is what? Nitrogen metabolism. Just this 5 concepts, if you learn it in detailed manner, then this chapter will be done. Okay. So today we are going to look into this chapter on whole, and tomorrow we will be exercising the EAPCET previous year questions. So look at here carefully, ma. Firstly, what is hydroponics? This is the technique of growing plants in a defined solution by providing minerals and nutrients in varied concentrations. When you supplement the solution with minerals and nutrients in varied concentration, you will get to know by the presence or absence of any minerals or nutrients, the growth of plant is controlled, and the deficiency symptoms that are observed in the absence of those nutrients will tell you the importance of each essential element. Who delivered this hydroponics first? It is first demonstrated by Julius von Sachs. It is first demonstrated by Julius von Sachs in the year 1860. Okay, the nutrient solution is aerated with optimum growth. Okay, and he is the first scientist who has developed, and he is the same scientist who said that glucose is the product that is formed at the end of photosynthesis. And this is about the hydroponics that I told you. And for the regular commercial production, we are using hydroponics for the development of vegetables, for the production of vegetables like tomato, seedless cucumber and lettuce. Whatever the climatic conditions might be outside, we can produce the plants using hydroponics. That is the major advantage. And here you have a look at the mineral elements that we have to know. More than 60 elements are found in different plants. Okay, we have more than 60 elements in different plants. Fine, right. More than 60 elements are found in different plants. Some plant species will accumulate selenium and gold as well. Some plants will grow near nuclear test sites. They have radioactive strontium inside them. There are techniques that detect the minerals even at very low concentrations. Low concentration, which means 10 to the power of minus 8 grams per milliliter. That much low concentration, if at all any minerals are present, we can detect them. Okay, so what is the criteria for essentiality of any element? What is the major criteria that we are telling that this is the essential element and this is not? Criteria for essentiality of elements, important. We can get statement based questions. Look at it carefully. An element must be needed for normal growth and reproduction. In the absence of this element, the plant do not complete their life cycle or set seeds. In such case, we can say the element is essential for the plant growth and development. The requirement of an element must be specific. It should be very specific. Means deficiency of any one of the element cannot be met by the another element. Okay, the element absence, any element's absence cannot be compensated by giving some other element. If the element is that specific, we can say that element is essential for the plant growth. Only 17 elements are absolutely essential for plant growth and metabolism. We have 17 elements like that, like nitrogen, phosphorus, potassium, magnesium, sulfur, calcium, boron, chlorine, manganese, ferrous, iron. Okay, nickel, copper, zinc, molybdenum, hydrogen, carbon, oxygen. Any of hydrogen, carbon, oxygen, you will get it through the raw materials directly. Okay. Essential mineral elements we have based on the quantitative requirements. Based on the amount of quantity the element is required, we have two types, macronutrients and micronutrients. Here macronutrients and micronutrients, when they are classified based on the macro, primary, secondary, non-mineral elements and mineral elements, and micronutrients. Here this table you can see clearly. If you want, you can take a screenshot as well. Here if you see carbon, hydrogen, oxygen, these are the non-mineral sources. Primary macronutrients like nitrogen, phosphorus and potassium. Secondary macronutrients like calcium, magnesium and sulfur. Micronutrients you can see boron, chlorine, copper, ferrous, manganese, molybdenum, silicon, sodium and zinc. These are the micronutrients. And then essential micronutrients. They are present in the plant tissues. Macronutrients, they are present in the plant tissues in bulk amounts like 10 millimolar per kg. Okay, 10 millimolar per kg of dry weight, if they are present, then that is addressed as what? Macronutrients. They include carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium, magnesium. Total 9 elements. Okay, 9 macronutrients in total. Now we are going to look into the trace elements. Trace elements, these are very small elements. They are present less than 10 millimolar per kg. They include iron, manganese and copper. Okay, they include iron, manganese, copper, molybdenum, zinc, boron, chlorine and nickel. These are the micronutrients or trace elements that are present. For higher plants, there are some other elements required such as sodium, silicon, cobalt and selenium. These are some higher elements that are present. Next, based on the functions, essential elements are of 4 categories. First is components of biomolecules and their structural elements of the cells. What are they? Carbon, hydrogen, oxygen and nitrogen. These are the structural elements of the cell. Next, energy related chemical compounds like magnesium, which is present at the center of the porphyrin ring. This is the porphyrin ring which is present in the chlorophyll pigment. Magnesium is present at the center of the chlorophyll, and phosphorus is the major component of the cellular energy, that is ATP. Next, elements that activate or inhibit the enzymes. What are the elements that are activating or inhibiting the enzymes? Magnesium, if you see, it is an activator for rubisco and phosphoenol pyruvate carboxylase enzyme. For both of these enzymes, magnesium is the activator. They are critical enzymes. There are some critical enzymes in photosynthetic carbon fixation. Next, zinc. Zinc is an activator of alcohol dehydrogenase and molybdenum of nitrogenase during nitrogen metabolism. Zinc is an activator for alcohol dehydrogenase and molybdenum of nitrogenase. Molybdenum is an activator for nitrogenase. This you can learn in the nitrogen metabolism. Elements that alter the osmotic potential of a cell. For example, potassium helps in opening, both opening and closing of the stomata. Next, role of macro and micronutrients. Essential elements, they will have role in their metabolic processes. What are the metabolic processes like? Permeability of the cell membrane, maintenance of osmotic concentration, electron transport systems. These elements will play crucial role in the electron transport system. Buffering action, enzymatic activity, constituents of macromolecules and coenzymes. Next, first is nitrogen. This is required by the plants where it is absorbed in the form of nitrates, nitrites, and it is also taken sometimes in the form of ammonia. Though ammonia is toxic to most of the plants, it will be converted to nitrates and nitrites mostly. Okay, and then here you see functions. What are the major functions of this nitrogen? See, major functions with respect to each element is important, ma. It is essential for all plant parts, mainly meristematic tissues and metabolically active cells. It is major constituent of amino acids, proteins, nucleic acids, chlorophyll, vitamins and hormones. For all this, nitrogen is the major constituent. Next is phosphorus. It is absorbed by plants in the form of phosphate ions. Phosphate ions and phosphite ions, you can see here. What are the major functions of it? Permeability of the cell membrane, and it is a major constituent of the cellular energy, that is ATP. It is also present in the nucleotides and in the nucleic acid, where phosphate is binded in the form of phosphoric acid. Okay, it is required for all phosphorylation reactions. Phosphate is required for all phosphorylation reactions. And then you see, next element is potassium. Potassium ions, these are highly required mainly by meristematic tissues, buds, terminal buds and axillary buds, leaves and root tips and shoot tips. At these regions, potassium is highly required because this maintains anion-cation balance inside the cell. Next, it is involved in protein synthesis, opening and closing of stomata. Both potassium and chlorine will play crucial role in the mechanism of opening and closing of stomata. Activation of enzymes, and this also helps in maintenance of turgidity of the cell. Okay, this helps in maintenance of turgidity inside the cell. Next is calcium. Calcium, it is absorbed from soil as calcium ions. Okay, calcium, it is absorbed from the soil as what? Calcium ions. It is required by meristematic and differentiating tissues. Okay, it is required both by meristematic and differentiating tissues. It accumulates in the older leaves. Functions: during cell division, it is used in the synthesis of cell wall, particularly as calcium pectate. And in middle lamella, it is also needed for the formation of mitotic spindle. Calcium ions, they will play crucial role in the calcium pectate and magnesium pectate. Calcium pectate, magnesium pectate, both is the composition of middle lamella, which is first formed wall layer in the plant cells. During the time of cytokinesis, what is the first formed wall layer in the plant cells? First formed wall layer is middle lamella, whose composition is calcium pectate and magnesium pectate. For normal functioning of the cell membranes, and it also activates certain enzymes, and it plays an important role in regulating metabolic activities. Next is what, ma? Magnesium. It is absorbed by plants in the form of divalent, like magnesium ions. Mg2+, magnesium ions. This will activate the enzymes both for respiration and photosynthesis. This is involved in the synthesis of DNA and RNA, and this is the major constituent in the ring of chlorophyll. You can also add on the concentration of magnesium ions will help in binding both the larger subunit and smaller subunit of the ribosome. The association of larger and smaller subunit of the ribosome is dependent on magnesium ion concentration. Okay, this is dependent on magnesium ion concentration. I hope this is clear for you. And then sulfur. Okay, sulfur, which is the major component in sulfur containing amino acids like cysteine and methionine, sulfur containing amino acids, whose functions, if you see, it is a constituent of several coenzymes, vitamins like thiamine and biotin, whose coenzyme A. Okay, and then if you see iron obtained in the form of ferric ions or ferrous ions, whose major functions, if you see, these are the major constituents of proteins, are involved to transfer the electrons like ferridoxins and cytochromes. You can see ferrous sulfur complexes in the ETS, electron transport system, both in the mitochondria and in the chloroplast. Ferrous sulfur complex, ferridoxins, these are present, right, whose major constituent is what? Iron. Okay, ferrous. It is reversibly oxidized from ferrous to ferric during electron transport. Okay, it is not irreversible reaction, this is reversible. It activates catalase enzyme, and it is essential for the formation of chlorophyll pigment. Then you see here manganese ions. This activates many enzymes which are involved in photosynthesis, respiration and nitrogen metabolism. Photosynthesis, respiration, nitrogen metabolism. These enzymes, okay, will be activated by what? Manganese, in the form of manganese ions. The best defined function is in the splitting of water, that is, water molecule will be split into protons, electrons and oxygen atoms during photolysis of water, which happens in the inner thylakoid membrane. Okay, that is because of what? What is the major role of the element there? Manganese plays crucial role during photolysis of water. Fine, right, ma? Next is zinc. The plant that obtains zinc in the form of as zinc ions. This functions, whose functions, like you see, it activates various enzymes, especially the enzyme carboxylases. Okay, this activates the enzymes like carboxylases. It is needed in the synthesis of the auxin hormone. Copper. This is absorbed in the form of cupric ions, whose major functions, you see, it is essential for overall metabolism of the plants. Like iron, it is associated with some more enzymes in redox reactions, and it is reversibly oxidized from copper to cuprous ions. 11th is boron. Boron is absorbed in the form of boric acid. Like it is required for what functions you see here? Uptake and utilization of calcium, membrane functioning, pollen germination, cell elongation, cell differentiation and carbohydrate translocation. These are the major functions. Next you see molybdate ions. Molybdenum is absorbed in the form of molybdate ions, whose major functions, it is a complex of many enzymes like nitrogenase and nitrate reductase. Both enzymes will come into picture during the time of nitrogen metabolism, which we are going to exercise now. Okay, these enzymes will participate in nitrogen metabolism. Next is chlorine. Chlorine, of course, this also plays major role in anion-cation balance, opening and closing stomata, and this is also helpful for water splitting reaction during photosynthesis.
Moving negatively charged elements. Cathode
Elements when they are not present. What are the deficiency symptoms that you see? Concentration of an essential element below the plant growth retards is termed as what? Critical concentration. Morphological changes due to deficiency or the absence of element. These such symptoms which are shown by the plant. The absence of the element. Deficiency symptoms. Okay.
partially charged setting, but
those deficiency symptoms that will depend on the that is movement of the element all across the plant. Elements that are actively mobilized,
energy
exported to young developing tissues. Deficiency symptoms
is required to eject the electron from.
You can see a healthy leaf, phosphate deficiency, nitrogen deficiency and potassium deficiency easily observed in the picture. So the deficiency symptoms of potassium and magnesium, they are easily visible, suppressed in the senescent leaves. Okay, remember this is a major statement directly from your text. Deficiency symptoms of essential elements. Older leaves where biomolecules containing these leaves are broken down makes these elements available for mobilizing to younger
Eject from the metal.
While they are not transported out of the mature organs, the deficiency symptoms are first appeared in the younger tissues. You have to be clear about these 2 points. Like the absence of what elements will show deficiency symptoms in the older tissues, absence of what elements will show the deficiency symptoms in the younger tissues. Where you can see symptoms in the younger tissues and where you can see in the older tissues. Both you observe it carefully. First you see in the younger tissue, if you see example, sulfur and calcium, they are a part of the structural component of the cell and hence they are not easily released. This aspect of mineral nutrition, they have great significance in agriculture and horticulture. Deficiency symptoms include chlorosis, necrosis, stunted growth, premature fall of leaves and buds and imbibition. Just go with the chlorosis and necrosis. Chlorosis. The loss of chlorophyll leading to yellowing of leaves. This is due to deficiency of the elements like nitrogen, potassium, magnesium, sulfur, ferrous, manganese, zinc and molybdenum. Absence of any of these elements will show the symptom of what? Chlorosis. And death of tissue. Particularly this occurs in leaf tissue. Necrosis occurs due to absence or deficiency of what? Calcium, magnesium, copper and potassium. Lack or low levels of nitrogen, potassium, sulfur, molybdenum, this will inhibit the cell division. Okay, low concentration of nitrogen, sulfur, molybdenum, etc. This will delay flowering. These are the deficiency symptoms that we are done till now. Next is toxicity. Excess of any element will lead to toxicity. See, for example, excess of manganese toxicity will lead to deficiency of iron, magnesium and calcium. Important. Thus, symptoms of manganese toxicity are nothing but deficiency symptoms of what? Ferrous, magnesium and calcium. Main symptom of manganese toxicity you can see is appearance of brown spots surrounded by chlorotic veins. Brown spots surrounded by chlorotic veins you can see. Next is mechanism of absorption of minerals. See, both apoplastic and symplastic pathway, we have learned it already. Both by apoplastic and symplastic pathway, water mineral ions are being extracted. At the region of root endodermis, which has suberin depositions, at that region apoplastic movement is inhibited and completely given to what? To plastic.
Like the electron ejecting
Clear till here?
lithium, sodium,
And then
calcium, and
nitrogen metabolism. See,
function is
first
they emit photoelectron when
the very first step is nitrogen is more prevalent enzyme and the limiting nutrient. The first step is the nitrogen will be fixed, which is nothing but nitrogen fixation. Nitrogen fixation is the first step in nitrogen metabolism, where the nitrogen is fixed by any of these processes, by lightning or by UV radiation or converting nitrogen to nitrogen oxide. By any of this, the nitrogen is fixed. And the second process is ammonification. The fixed nitrogen is converted to that is like you said, decomposition of organic nitrogen of dead plants, like decaying biomass of dead and decaying plants and animals. This will be converted to what? Ammonia. Nitrogen converting to ammonia is known as ammonification. So the very first step is what, ma? Nitrogen fixation. From this image you see. Yeah. First you observe here, first is what? Nitrogen fixation.
Mass into
velocity, where nitrogen is fixed either through biological nitrogen fixation, industrial nitrogen fixation, electrical nitrogen fixation, by any of these, fixed in the form of nitrates, nitrites and ammonia. Next, this ammonia is converted to nitrates and then to nitrites. So once nitrogen is fixed, nitrogen converting to ammonia, the second step is what? Ammonification. Third step is nitrification. Ammonia converting to nitrate is by nitrosome and nitrosococcus. Nitrosomonas and nitrosococcus. And second, nitrate converting to nitrate.
Like what?
This is through what? Nitrate converting to nitrite. This is through the bacteria Nitrococcus and Nitrobacter. Nitrococcus and Nitrobacter. Here you see, first ammonia converting to nitrate, that is by what? Nitrosococcus, Nitrosomonas. Next is Nitrobacter and Nitrococcus. You can see these steps are called as what? Nitrification. Nitrates are formed as such. Now, next is denitrification. Denitrification. Denitrification is done by sulfur containing bacteria like Thiobacillus and Pseudomonas. Okay, Thiobacillus and Pseudomonas, it will do the fourth step. Okay, that is denitrification. Here the plants will absorb nitrate and they transport to leaves. In leaves it is reduced again to ammonia. Ammonia here in the leaves should be converted to what? Amino acids for the proper assimilation. Now last step is what? Assimilation of ammonia to amino acids. How the step is being done, you see. How ammonia how denitrification is done. Nitrates and nitrites after ammonia formation, how ammonia is being assimilated in the form of amino acids. This is by two steps mainly. Why this assimilation is done? Why ammonia have to get converted to amino acids? This is most of the plants will convert nitrate to nitrate and ammonia, but ammonia is quite toxic to few plants. So they have to be converted to amino acids for proper assimilation. Here you can see the first step is what? Reductive amination, and second step is what? Transamination. Reductive amination and transamination. Reductive amination and transamination. In this, ammonia will react with what? 5 carbon compound
Chapter
alpha ketoglutaric acid. You can clearly see this over here. Here ammonia is reacting with alpha ketoglutaric acid by consuming the energy NADPH and the enzyme glutamate dehydrogenase. Here then it will be converted. Next step is what? Glutamate. Alpha ketoglutaric acid, when it is reacting along with ammonia in the presence of the enzyme glutamate dehydrogenase, this will be converted to glutamate, water, and NADP plus is leached out. Here we go with transamination. This is to transfer amino group from one amino acid to keto group of
the capacitor plates is
this is by the enzyme transaminase. The enzyme transaminase, this will help in the transfer of amino group of one amino acid to keto group of the next keto acid. Glutamic acid is the main amino acid from which the transfer of ammonia takes place, and another ammonia forms through the process of transamination. Here at last, asparagine and glutamine are the two most important amino acids which are found in plants. They are the main structural part of the proteins. Here we have
is double
amino acids. Those are aspartic acid and glutamic acid. So we have two steps. One is
HC
reductive amination,
HC by
process is transamination process. Okay. And here one thing,
magnetic radiation is
nitrogen fixing bacteria. We have two
this is the
bacteria. One is
E dash will be
free
C by 2 lakh.
fixing bacteria and the second is
So what we
fixing bacteria. Here you can go through free living nitrogen fixing bacteria like
is double
Bacter, Bacteria, which are aerobic free living nitrogen fixing bacteria. Rhodospirillum and Bacillus, these are the anaerobic nitrogen fixing bacteria species. Next, cyanobacteria are like Anabina and Nostoc, these are nitrogen fixing free living bacteria. Anaerobic and
give
Symbiotic nitrogen fixing is Rhizobium leguminosarum. Rhizobium bacteria, which is a gram-negative bacteria, which will fix the nitrogen symbiotically
and to observe
with legume plants. This is in symbiotic association with legume plants. Legume bacteria relationship. Here, Rhizobium species in roots of the legume plants like alfalfa, sweet clover, sweet pea, lentils, garden pea.
And when
these plants, Rhizobium is fixed. Okay, that is why the family bean family is also addressed as Leguminosae. Okay, Fabaceae or Leguminosae. Next, biological nitrogen fixation. One more bacteria, that is Frankia, which is in symbiotic relationship with a plant like apple. Rhizobium and Frankia are free living in soil, but they also act as
boards.
Rhizobium with legume plants, Frankia with alnus. Okay. Remember that carefully. What is the major procedure here? Internal procedure that is happening during the process of nitrogen fixation. Bacteria which is associated into the soil particles, it gets aligned with the root, where the root forms the hook-like structure when bacteria close to it by forming what? Infectious thread. Bacteria will form an infection to the root hair, which forms an infectious thread, and later this matures to a mature nodule. Okay, and that gives pink color because we have learnt nitrogen fixation is in the presence of the enzyme nitrogenase. Nitrogenase is sensitive to oxygen. Remember, nitrogenase is sensitive to oxygen.
Automobiles.
Enzyme
Radar
is sensitive to oxygen. Fine, right? This is sensitive to oxygen, and this nitrogenase is functional in the anaerobic
aircraft.
As such, some pink globules are formed to give that anaerobic structures, okay, anaerobic regions during the time of nitrogen fixation. Okay, and in the presence of nitrogen, nitrogen fixation is an energy requiring process. Here you see nitrogen converting to ammonia will require 16 ATP for the formation of 2 ammonium ions. 16 ATP is required. which is through biological nitrogen fixation. Fine, right? So these are the enzymes. Okay, like hemoglobin acts as what? Oxygen scavenger, because in the presence of the oxygen, nitrogenase will be inactive. So like hemoglobin will act as oxygen scavenger, which will give anaerobic conditions, which will help the nitrogen fixation to happen though oxygen is present around the conditions. Clear till here? So this is all about this chapter. So what all we have learned in the beginning about Julius von Sachs, who has first demonstrated the process of hydroponics, followed by hydroponics, how is it important, statement based we have looked into, and then about essential elements, toxic elements, and then about toxicity, and then about trace elements. Most importantly, deficiency symptoms in the absence of macronutrients and micronutrients. And then we have looked into nitrogen metabolism in detail. How mineral ions are being uptaken also we have seen. And lastly, we have also looked into the physiological process that is happening during nitrogen fixation and complete process of nitrogen metabolism and the bacteria which are involved for ammonification, nitrification and denitrification processes. All these concepts are the major concepts of mineral nutrition. In our next, we will exercise substance of mineral nutrition, and then we will go on to photosynthesis in higher plants. Okay, that's all for today's session, and thank you.